Bismillahirrahmanirrahim. Assalamualaikum. Dear students, class 9th biology. आज हम चैप्टर नंबर 5 सेल साइकिल का अगला टॉपिक मियोसिस डिस्कस करेंगे. मियोसिस इज़ अ लॉन्ग क्वेश्चन इन चैप्टर नंबर 5 और इस टॉपिक से कुछ शॉर्ट क्वेश्चन और MCQs भी सिलेक्ट किए जा सकते हैं. पहला शॉर्ट क्वेश्चन जो इस पेज पर हमें नज़र आ रहा है, दैट इज़ द डेफिनेशन ऑफ मियोसिस. डिफाइन मियोसिस और व्हाट इज़ मियोसिस. Meiosis is the process by which one diploid eukaryotic cell divides to generate 4 haploid daughter cells. दूसरा सवाल, what is the difference between diploid and haploid? Diploid means cells in which chromosomes are in pairs, while haploid means the cells with half the number of chromosomes, or they are not in the form of pairs. Some MCQs from this page are, the first one is from the definition of meiosis, other one from diploid and haploid. And the word meiosis comes from the Greek word meion, meaning to make smaller. Meiosis is reduction in chromosome number. There are two short questions on this page. The first one is about the interface of meiosis. Interface of meiosis is divided into three phases, G1, S and G2. Why meiosis 1 is called reduction division? In meiosis 1, the homologous chromosome in a diploid cell separate and 2 haploid daughter cells are produced, so it is called reduction division. Some MCQs may be, the first one is about the discovery of meiosis. Meiosis was discovered and described for the first time 1876 by Oscar Hartwig. In meiosis 1, 2 haploid cells are produced. The next question is, what are the two main steps of meiosis 1? Those are karyokinesis and cytokinesis. In what phases the karyokinesis of meiosis is subdivided? Karyokinesis of meiosis 1 is subdivided into prophase 1, metaphase 1, anaphase 1 and telophase 1. The next topic is prophase 1, that can be selected as a long question separately as well. prophase 1 में यहाँ पर कुछ शॉर्ट क्वेश्चंस मौजूद हैं. पहला शॉर्ट क्वेश्चन जो है, दैट इज़ अबाउट synapses. Homologous chromosome line up with each other and form pairs by a process called synapses. The other definition is called bivalent. Each pair of homologous chromosome is called bivalent and it has 4 chromatids, so it is also called as tetrad. तो ये क्वेश्चन भी पूछा जा सकता है, what is the difference between bivalent and tetrad? What is chiasmata? The two non-sister chromatids of homologous chromosomes join each other at certain points along their length. These points of attachment are called chiasmata. And the last question is about crossing over. The non-sister chromatids of homologous chromosomes exchange their segments and this phenomena is known as crossing over. The MCQs are from all the definitions which we have discussed. The first one is about karyokinesis is subdivided into 4 phases. Prophase is the longest phase in meiosis. The others are from definitions: synapses, bivalent, tetrad, chiasmata, and crossing over. The next question is, what is the contribution of Morgan? In 1911, the American genetist Thomas Hunt Morgan, T.H. Morgan, observed the phenomena of crossing over in fruit fly Drosophila melanogaster. The next question is about the metaphase 1. What is metaphase 1? During this phase, pairs of homologous chromosomes align along equatorial plane, forming the metaphase plate. The MCQs may be, the first one is, the exchange of segments result in the recombination of genetic information. After crossing over, each pair of homologous chromosome remain as a bivalent. When nuclei disappear during prophase 1, the nuclear envelope disintegrates. In mitosis, two kinetochore spindle fibers attach with one chromosome. The next one is, what is anaphase 1? During anaphase, kinetochore spindle fibers are shortened that result in the pulling apart of chromosomes. One chromosome is pulled towards one pole and two haploid sets are formed. What is telophase 1? During telophase 1, chromosomes arrive at the poles, and each pole having half number of chromosome consists of two chromatids. Spindle network disappears and the nuclear envelope is formed around each haploid set. Cytokinesis of meiosis 1. Animal cell has the same process cytokinesis as was in mitosis, while plant cell also have the same. Some MCQs may be, during anaphase 1, kinetochore spindle fibers shorten. During anaphase 1, two haploid sets of chromosomes are formed. During telophase 1, haploid set of chromosome is formed. Cytokinesis is the pinching of cell membrane in animal cell and the formation of cell wall in plant cell. The next one is about meiosis 2. What is interkinesis or interphase 2? After meiosis 1, both haploid daughter cells enter a period of rest which is known as interkinesis or interphase 2. How interphase 2 is different from interphase of mitosis and meiosis 1. The interphase 2 is different from the interphase of mitosis and meiosis 1 because there is no S phase, and so there is no duplication of chromosomes during this stage. The next question is, in how many phases meiosis 2 is divided? Meiosis 2 is divided into prophase 2, metaphase 2, anaphase 2 and telophase 2. The MCQs may be, prophase 2 takes much less time as compared to prophase 1, and the other one is, meiosis 2 is similar to mitosis. And the last one is, what is the result of meiosis after telophase 2? The result is nuclear envelopes form. Cleavage or cell wall formation eventually produces total of 4 daughter cells, each with a haploid set of chromosomes. So dear students, today we have discussed the process of meiosis with reference to long questions, short questions and MCQs. Inshallah, in the next topic we shall discuss some other topics of chapter number 5, cell cycle. Till then, Allah Hafiz.